Hey guys, so welcome to Subu Medicine, and this is Immunology. Today we'll be discussing about the T lymphocytes, alright? That's what we're going to talk about. And let's start. So guys, the first thing that we have to discuss about here is that, where do these T lymphocytes actually born, alright? That's what we have to talk about first. So, guys, if you know guys about T lymphocytes, right, from the bone marrows, these guys are born in, basically these guys are born, the T lymphocytes, right? These guys are part of the lymphoid series, all right? So, if there is something called like hematopoietic stem cells. Let me just write down here, right? Okay, we'll just make it right here actually, in the bone marrow. There's something like hematopoietic stem cells, right? And what is going to happen here is that these hematopoietic stem cells will divide into two lineage. One lineage is going to be, let's say, the lymphoid lineage, all right? Lymphoid lineage. And the other one is going to be myeloid lineage, right? So, from this stem cells, right? This is let's say hematopoietic stem cells, right? From the hematopoietic stem cells, based on the cytokines, all right? And you gotta remember this, like either it's interleukin number 7 and then interleukin number 3. Remember, how the heck the stem cells know whether they are going to go to, make lymphoid cells or they're going to make myeloid cells. Remember, myeloid cells are what? Those are red blood cells, platelets, those are white blood cells like neutrophils and going to be, eosinophils and basophils. And whenever we talk about lymphocytes, we talk about the B cells and T cells and the natural killer cells, right? So how do you know that the stem cells know whether they're going to go to whether they're going to make the lymphoid or they're going to make the myeloid cell? And that is based on these interleukins, okay? If there is a secretions, and these interleukins are cytokines, right? If there's a stimulations of the interleukin number 7s to this hematopoietic stem cell, they're going to go and make the lymphoid cells, like T cells and B cells. If there's a stimulations of interleukin 3, they're going to make the myeloid, those neutrophils, right? Or basophils and eosinophils. So that's something that you have to remember, right? So let's say because we're discussing about the lymphocytes, primarily we're focusing on the T cells, so that's what we have to focus on. So these hematopoietic stem cells, whenever there's stimulations of the interleukin number 7, they'll take the lymphoid series, okay? And in the lymphoid series, what is going to happen? This is a lymphoid lineage, okay? And ultimate result here, what happens is that these cells will make something called prolymphocytes, all right? Prolymphocytes. And these prolymphocytes will make this as a prolymphocyte right here. If I make this as prolymphocytes right here, right? These prolymphocytes will divide into two parts, all right? Either it will go and make this cells right here, or they'll make another cells, which is going to be let me make this with this one. Either with this cells right here, all right? And we can simply say this is okay, this is going to be my B cells, because this is what it's going to form, let's say, B cells. So if you want to be very, very correct, these are called, pro B cells now. And these are called pro T cells, right? This is what's happening now. We do not know exactly why, or what stimulates this productions of your, what stimulates to, differentiate into B cells, or what stimulates differentiate into pro T cell. Like, at least I do not know, right? If you know, just let me know, right? But the point being here is that there is going to be interleukin number 7s, but at the same time, there are other growth factors to remember. These guys are happening in, in the bone marrow house, and bone marrow has this stromal cells, okay? They're providing some of the growth factors, like, for example, the stem cell factors like that. There are other growth factors, like, for example, like c-kit, all right? The notch, ligand, all right? All of them are like also participating, or they're also nourishing the cells, right, to proliferate or to differentiate it into more mature cells. Because right now, these guys are at early phase, all right, and they're immature cells, all right, and you need all this growth factor in order for them to differentiate or proliferate, all right? Now, we're focusing the pro T cells right now, right? Now, in this process, pro T cells right here, let's make this pro T cells right here. Let me make this pro T cell and more looks like, let me make this a T cells right here, right? So it looks like a T cell for you, all right? Now this is a T cell, right? All right? So these T cells right now, we call this as a pro T cells. Look, what do we call this? We call it a pro T cell. In the pro T cells right now, what is going to happen? These pro T cells right now is, there's nothing is there, right? There's nothing is there. There's no markers. There's nothing there, right? So it is a very, very early stage of this, the pro T cell, And what is going to happen with the pro T cell? Once this pro T cells are synthesized in the bone marrow house, and remember, this is usually like, taking place in a very early, gestation period, like 8th week of the developmental period, right? This is what it's happening right here. And what is going to happen here is that this pro B cell, what they do, they will go and migrate into this area right here. And what is this area right here called? This area is what is called as your thymus. This is where your T cells will get mature, or this is where their selection, or this is where there's a concept called central tolerance, maturation selections. This is where, it will take place. Remember, T cells they're born in your, let's say, in a bone marrow house, right? These immature T cells will migrate to the thymus and they will get trained and select and they will become mature there, right? So that is what we have to discuss here right now. And this is in this process, during the maturations, the process, this is where they also developed like your receptors for them, all right? Now, when they are this pro T cell stage, right, what is going to happen here is that they'll migrate. But remember, why the T cell does not migrate to other parts of the area? Because there are other areas also. So the blood, when this pro T cell migrates into it, what is going to happen is that there are different types of growth factors here, right? And also, if you look at the thymus is a multi-lobe structure, right? It has a cortex, it has a medulla, right? It has that. And this cortex right here, especially this cortex area, they have these epithelium cells. These are, it has like pseudopod structures. And these guys have, do you know where they're originated from? From the stromal cells, right? So the bone marrow also provides the, environment for the T cells to actually, work within that microenvironment that has the stromal cells, and those cells provide growth factors. And these guys also, this epithelium, we call them a thymic epithelium, right? These thymic epithelium also provide that sort of like a microenvironment for the T cells, right? So these T cells, like when these pro T cells, right, when they get migrate to, let's say, this guy right here, right, in the cortex, what is going to happen here? Before we go talk about this maturation and selection process, let me tell you something. All the maturations, all right, or selection process, that is typically happen in the cortex area, right? And one more thing that I do have to talk about here is that in the cortex, you also see some of the corpuscles like this, right? These corpuscles are called Hessel's corpuscles. And do you know what signifies this Hessel corpuscles? They signifies the derivatives of thymus. Thymus are made out of those third pharyngeal arch, right? Those when those third pharyngeal arch come together, they will what? They'll make this what? They'll make this thymus, right? So this Hessel corpuscle that you get to see, which is located in the medulla of the thymus, these are the derivatives of like those third pharyngeal arch, all right? Now, after this, a little bit of the anatomy of the thymus, right? Now, when the protein cells, right, from the blood, when it goes to the thymus, like right here, let's say this thymus is right here. So the T cells. And these T cells, we call this right here, thymocyte, right? The thymocyte, why? Because the T cells are what? T cells are dependent on thymus, right? Thymus cells. And this T is usually referring as a thymus, right? Thymus dependent T cells, right? And this thymus we also called a throne of immunity. They also call this as a throne of immunity. All right. Now, when this pro T cells comes right here, right? Let's do this. What is happening here is that, you know, this is this pro T cells right now, right? What is it? If you see that these T cells are naked right now because they have nothing on the surface of them, right? So when they have no surface of them, you know, what do we call them as we call it something called, you know, double negative thymocyte. What does this actually means? You know, what is the concept of double negative thymocyte is that, you know, there are usually, you know, let me just quickly tell you. So let me just quickly tell you about this. What happens is that if you look at this right here, you know, if let's make it simple, if this is my thyme, if this cells right here, right, if this shows something called like the CD4 positive and then CD8 positive, right? Because usually, you know, what happens is that if you look at the bigger picture, your T cells primarily divide into the two main things. CD4 cells or CD8 cells, okay? The CD4 cells, we typically call them helper cells, and CD8 cells, we call them a cytotoxic cells, all right? So based on this marker, we either tell them, okay, if your T cells, okay, if they have both the markers, CD4 plus CD8, then we call them that because it's 1, 2, first, second, right? So they're double positive. All right. If they don't have this both marker, then we call them a double negative, right? So because these are showing there's no markers of CD4 and CD8, then we call them these are double negative cells. This is completely depends on these markers, right? Because in this process, what is happening here, this selection process that's taking place in the cortex of thymus, this is what is called central tolerance, right? What is this called? This is called central tolerance. Basically, they are going to go something called positive and negative selections, and we'll talk about that, right? But the concept here is that this double negative thymocytes is based on the marker. These are naked T cells. They're immature cells, right? These immature T cells, right? So when they guys come right here, you know what is going to happen here? Look at this epithelium, this thymic epithelium. They'll secrete a lot of this, like, you know, growth factors, all right? Or something called, like, you know, there's something called thymosin, all right? Thymopoietin, all right? You know, other like a growth factors to like notch. Okay, and what happens with this? Under the influence of this hormone, what is going to happen is that, you know, they will stimulate this double negative thymocyte, right? And when that happens, right, what is going to happen is that will lead into the activations of like a certain genes into this your double negative thymocytes, because right now they don't have anything. They also don't have your T cell receptor, right? TCR. Remember, we need to know a couple of things, okay? We need to have T cell receptors, one, and this T cell receptor needs to express on the surface. And after that, we need to also have CD4, we need to know, and then CD8. And obviously, all right, before that 3, we'll write down here 3, we have to have signal transductions, and then we need to know whether cells are CD4 positive or let's say CD8 cells, all right? So these are the steps that we need to know, all right? So right now, these guys do not have any markers. Under the influence of this, right, what is going to happen here is that this could activate certain genes in this double negative thymocytes, right? Now, in this, or you can say pro T cells right now, right? When that lead into activation certain genes, what is that going to happen is that they're going to synthesize something called T cell receptors. So now we have to know how this T cell receptors are synthesized, all right? Now, for that one, you have to come back here, right? You know, under the influence of this, like hormones and growth factors, what is going to happen here is that, there is a activations of like in a chromosome number. Let me write it down here. Because remember, it's a chromosome number 14, all right? There is a activations of gene on the chromosome number 14, all right? And what is that going to lead into? That is going to lead into synthesis of something called alpha chain, okay? Alpha chain. And this alpha chains are light chain, all right? Now, before I go detail about this, let me show you receptor. This is how your receptor looks like, your T cell receptor. If you look at this receptor, look, you have this right here. This is an alpha chain right here, right? And this is a, let's say, beta chain right here, right? If you look at the beta chain, there are different segments here, right? This portion right here, okay? This portion right here, we call them it's variable regions, okay? In the alpha chains. And this portion right here, this is joint regions, right? And then you have a constant regions because that's going to stay constant, right? And then you also have this tail that is actually passing because this is located outside of the plasma membrane and this is located inside the plasma membrane. And you can call this right now, which is located now, this is facing the cell, this part. We call them a cytoplasmic tail, if you want to remember this, all right? Now, and if you look at the beta chain right here, you also have variable regions, okay? You're going to have J regions, obviously joint regions. You have a constant regions, but at the same time, in the beta chains, okay, they're also called heavy chains. They also have something called diversity segment. This is the reason why, T cells are very, very important because of the diversity, that can help us to identify or bind into lot of different types of variety of pathogens, all right? So that, because they synthesize the diversity, it helps with making something called this whole concept of the, recombinations, okay? They can make multiple binding site for any pathogens or multiple binding sites where they can actually identify those viruses or bacteria so they can kill them, all right? So this is just if you look at this, this is how your T cell receptor looks like. Now we have to talk about how these T cell receptors are actually synthesized, all right? If you look at here, if I say the chromosome number, okay, chromosome number 14, they have a gene, okay, or specifically locus, they have a gene to synthesize this alpha chain, and alpha chain leading into synthesize our light chains, which are smaller, right? And the way it works is that, okay, when they're synthesizing, right, there's replications happen, and here I'm showing you this is a RNA, all right? If you look at this 5 and 3 prime body, right, I divided this into different fragments. Let's say this fragment right here, this fragment is my variable regions, and this fragment right here, okay, this is going to be my joint regions, and this is my constant regions, right? So usually, this segment right here, this variable region segment, they usually code for about, right? This is usually code for about 45 variable, okay? And right here, this joint, they code usually for 55 joint, and then if you say this, the C, usually it's one constant, right? So if you multiply this 45 times 55 and 1, what do you get? You get about 2,475, okay, combinations. Because remember, whenever you do 1 here, you do 1, 1, 1. That's how you make one alpha chain, right? One light chains. So if you're making the 45 of them, when you multiply 45 and you're making a 55 Z, which is a joint complex, all right, and you're making the one constant, if you multiply them, you can able to get 2,475 combinations, recombinations, all right? It is a very important concept. And this is basically showing the diversity of it, okay? And also the concept of this clonal expansion, all right? Now, I'm going to come back to this, all right? If you look at this one right here, all right? This long, this is referring as a heavy chain, all right? Because it's bigger, all right? And this is referring as a beta. Beta, right? It's a very important concept. And usually, to synthesize the beta chain, there is a chromosome number 7. Okay? In the chromosome number 7, there is a gene that can actually encode for the beta chains to make a T-cell receptor, okay? And even here too, there is going to be variable regions right here, okay? But one thing that is different in the beta chain here is that they have something called diversity regions, they have a joint region, and they have a constant regions, all right? It's very important. And now, we have to know, in this complex, right? In this complex, how much, like, let's say, what's the segment here on the variable regions, okay? Usually here, they encode for like 50 right here, okay? And there's a diversity, which is encode for like 2, and this encode for like about 12, right? And then you have a 2. So this is a 2 constant, 12 joint, okay, sequence, and then there are 2 diversity, and then a 50. When you multiply this, right, you should be able to get 50 times 2, 12, 2, you should be able to get 24, 24, 48, 48 times 50, 48. You should be able to get about like 1200 beta chain combinations, right? Now, so in order to make a TCR receptor, right, usually TCR receptors there are 2 kinds, okay? One of the most abundant that are present in our body are made out of alpha, this one, which is a chromosome of 14, and then chromosome number 7, which is a beta, alpha and beta chain. They're most abundantly present in our systems. There are also one other more receptor, which is produced by, this is called delta and gamma. So, okay, this is going to be delta here, and this is going to be gamma, right? There is a T-cell receptor, which is delta and gamma. They're usually located in intestines, right? Not most abundantly present, but there are other types of receptors that are present. And they also follow similar pattern right here, okay? So if they're making a delta right here, right? So usually it's like a 2, 3, right? 4, and then you're going to have 2, 3, 4, and then you have a 1. So basically you have 2 variable regions, you have a 3 diversity regions, and then 4 right here. This 4 is going to be a joint regions, and 1 is a constant regions. All right? And if you're talking about this one right here, which is a gamma, you have a 5, and then 1. Basically here you have a 5 variable regions, 5 joint regions, and 1 constant regions. All right? Now, this one right here, this is present in the, let's say, chromosome number 14, and this is chromosome number 7. This is going to be heavy chains, and this is going to be a light chains, okay? And this is just extra I'm giving it to you. But one thing that I do want you guys to remember here is that, okay, I'm telling you to know that how these T-cell receptors are synthesized, okay? The chromosome number 14s and chromosome number 7. Just know that they are important for synthesizing your alpha chains and beta chains, okay? And they will synthesize something called these T-cell receptors, all right? Now, but there is one concept here which you guys have to remember, all right? The concept here is that, during this process, right, this recombinations is happening, right? But remember, in this concept, what is happening here is that, how this recombining, all right? The sequence, okay, of this, let's say, this different sequence of this, different sequence of the genes, right? Whenever you do that, right, you need to have an enzyme that actually do the functions or the role of the recombination, or in order to do recombine, you also have to maybe do some kind of like alternative splicing. You have to break down, you have to cut down, and you have to rejoin them, all right? And so that way, you can able to, make a one good alpha chains and one good beta chains, all right? And for that, the special enzyme is required for that, all right? And there is an enzyme. That enzyme which does splicing, okay, and do recombination, we call them a VDJ recombinase enzyme. And you need to remember this, all right? VDJ, variable diversity joint recombinase enzyme. And And or there's another name they do give this, which is called RAG. RAG1 and RAG2. What does this mean? RAG is referring as, you can say rearrangement activating gene, or you can say recombinations activating gene. Because they are basically, combining, making a change, and they're activating the genes, all right? Because sometimes what happens is that if there's a deficiency on this recombinase enzyme, this RAG1, what it's going to lead into is that you're not going to form alpha genes. You're not going to make a beta genes. If you're not going to make a receptors, you're not going to recognize, if there's no receptor present, you're not going to recognize any sort of pathogens or bacteria or antigens, right? In that case, what is going to happen? You'll be immunocompromised, right? So there is whenever there's a deficiency on RAG1, RAG2, there is a condition called, you can get Omenn syndrome, all right? And this is usually associated with something called SCID. What is this? Severely compromised immunodeficiency, all right? Now, after this, I also have to talk to one more thing. Not only there's a RAG1 and RAG2 enzyme there, but there's also another enzyme. What is that called? Which is called TDT. What does this mean? You have a terminal, all right? There's a terminal and deoxynucleotide transferase enzyme. What is this importance of this terminal deoxynucleotide transferase enzyme, or TDT? Look. RAG enzyme, they do recombine, reactivated the genes, recombine, activated, splicing down. They did that, right? They did the amazing part. But then remember, sometimes what happens is that, there could be an extra, let's say, nucleotide is present, right? They're not supposed to be there. This is where the TDT comes in. The TDT comes in, removes that, or replaces some other, like nucleotide, and it what? It rejoins them, all right? So basically what the TDT do is that they're creating something called, you can simply say this is called, joining complex, or you can say this is a junctional complex they're making, all right? The joining complex, this TDT is doing. If there is deficiency on this, that would not result into that, right? So there's an important concept of this TDT enzyme, which is a terminal deoxynucleotide enzymes, all right? And usually this one, TDT usually we use as a clinical marker for the immature T cells, like pro T cells, all right? So it is important that you know, all right? Now, in order for the T cells, you need alpha and beta genes, right? But this is not enough. They also need something called signal transduction molecule. Look, first here we talked about, first thing is going to happen, rearrangement of the T cell receptor. That needs to happen, right? So this is the rearrangement part right here. We talked about this, right? Now, after the rearrangement of T cell receptors, there's also like what is going to happen? There's a gene that is going to, encode, signal transduction molecule, all right? And that signal transduction molecules, not going to go super detailed about it, but to know that there are going to be 3 of them, all right? Let me just write down, there are going to be 3 of them. Okay, let me just write down 3 of them. Because there are going to be 1, 2, 3, right? These 3 of them are there. Because of that, we call them a clusters of designation 1. This is called cluster of designation number 2. And this is called cluster of designation number 3. It's because that 3 are present, these are signal transduction molecule, we call them a CD3, all right? CD3 positive. That is the reason why all the T cells, all the T cells have something called CD3 positive. Even if it's a C4 positive, it's a CD8 positive cell, they're all gonna show something called CD3 positive. All right? This is how we call them CD3, all right? Now, after this, there's also if you want to remember extra, there's also something called in here, there's also another transduction molecule which is called Zeta. Zeta transduction molecule is also present there, all right? All of them. You have this part right here, alpha and beta chain right here. You have a CD3 positive right here, and also you have delta. You have this Zeta chain. They're called Zeta chains. All of them lead into it. All of them make this whole complex of this TCR receptor with the signal transduction molecule. Because these guys, CD3 positive cells and Zeta, they're playing roles as a signal transductions. All right. It is a very, very important concept right here. Now, after this, right, Now this cell, all right, they start synthesizing, right? What is going to happen here is that, okay, I'll make a simple right here. This guys, okay, I'll make a different cells right here, okay? What is going to happen, these are my T cells right here. You're going to have a marker right here. This marker is started showing, all right? They're showing this marker. You know, not going to go super detail about it, but like just letting you know, there is going to be first synthesis, a heavy chain first, and then the later light chains will start synthesizing a little later, right? So that's just letting you know. But for now, just know that, okay, they started showing this receptor now. And what is this receptor? You are TCR receptors, all right? Now, after this, all right, now the TCR receptors is there. Now we have to also talk about, all right, okay, we can say this, can start saying CD3, positive cells are. And now what is going to happen is that, you know, these guys with epithelium, they will, you know, they will show something called self-antigens. They will show some peptide to these T cells. All right, they'll show that. And what is going to happen here is that this thymic epithelium, you know, they show something called MHC1 and MHC2. What is this? It's called major histocompatibility complex, okay? Or you can also call them as a human leukocytes antigens. It is a very important concept here, guys. These MHCs are also called human leukocytes antigens, all right? There are going to be 1 and 2, all right? And if they are human, these are, let's write down HLA, right? If they belong to MHC1, then we usually, you know, classify them as like, okay, it's going to be HLA, HLA-A, HLA-B, HLA-C. And if this is belong to, let's say, MHC2, we say HLA, we say DP, HLA-DQ, and HLA-DR. It's important to know this, is because there are a lot of, like, you know, whenever we talk about disease, you know, they're going to often refer them as in HLA, DP, DQ, and DR, right? And these D, A, B, these are based on the isotype, okay, based on the alleles that are present in there. Whenever we're talking, there's a concept of morphogenic, okay, based on, which means that the poly means many, right? The many genes, you know, and because of their variability, you know, and we all have a different MHC1. You have different MHC1, I have a different MHC1, all right? They're made out of different sequence of the amino acids. Is because of that, there is a variability in each one of us, all right? So you have to remember, whenever you say one, just think about it's ABC. But whenever you think of MHC, think about DP, DQ, and DR, right? And all of them has a deep D into it. And what is the importance here is that the MHC1, right, it is present in all the nucleated cells. Some of the cells that are not nucleated, like red blood cells, platelets, MHC1 is not shown, all right? Except from those two, all the nucleated cells will have it, nucleated. And these MHC2s are usually, you know, shown by, let's say, these are thymic epithelium. These are shown by antigen-presenting cells, like, for example, macrophages, dendritic cells, B cells, all right? And even obviously thymic epithelium, like a fibroblast, they'll also show that, right? The vascular endothelial cells will also show the MHC2. But if you want to remember something, right? MHC, always remember that thymic epithelium will show it. They have that. This medullary epithelium will have it. All the antigen-presenting cells will have MHC2. Specifically, if you remember antigen-presenting cells, dendritic cells, and your B cells and macrophages, they will have MHC2, right?